I'm
mission.
just, yep. Okay, awesome. So we'll let other people trickle in, but I'm going to go ahead and get us started for timing. So it is my distinct pleasure to introduce Dr. Rohit Singh today. Dr. Rohit Singh is an assistant professor at Duke University in bioinformatics and cell biology. His research interests are broadly in computational biology, ranging from protein design, which is kind of what our seminar is known for, but also things like small molecule drug discovery, single cell, and also causal regulatory inference. He is super interested in probing biological foundation models to better understand the biology that they capture. He is a recipient of Duke University's Whitehead Scholar Award, the Test of Time Award at RECOMB, and MIT's George M. Sprowls Award for his PhD thesis, which he did in computer science, and Stanford's Christopher Stephenson Memorial Award for his master's research in the same field. Before he started his journey in academia, he also had a lot of experience in quantitative finance. So, Rohit, we're super excited to have you today. We're super excited for your talk. So, please take it away whenever you're ready.
Thanks, Meg. Thanks, Leo. That was a wonderful introduction. Thank you for that. Hi, folks. So this is me going to be sharing some fairly recent thinking about some of the work that we have been doing and some underlying themes that seem to me to be emerging. So I'll start off by sort of giving you a bit of context of our lab. As Meg mentioned, we do a lot of things. And so, but there is some sort of structure to what we're interested in. And essentially, we are first and foremost interested in trying to explain biology. And in that, we are interested across scales, like at the molecular scale as well as the system scale. And every once in a while, if you can explain something well, you can control it as well. And that will lead to interesting projects on its own. And that sort of is how things work out for us, where we have a bunch of things where we essentially are trying to model something at the molecular level. Sometimes when we do that, we end up understanding something or have an insight which can lead us to a design or a control. And for the most part, in this talk, I'll stick to the molecular scale, but there will be a story about something at the system scale. The other overarching sort of theme through what we work on is what makes these things possible, and what makes both sort of all the dimensions we look at possible essentially boils down to the sequencing revolution, where over the last 20 odd years, the cost of sequencing a genome has come down from about $1 billion to about $100. And that manifests in two ways. One is we just have a lot more species we have sequenced. So we just have a lot of protein sequencing data or gene sequencing data. And it also manifests in us being able to do a lot of single cell sequencing, where for an individual or an organism, we can just look at their individual cells and essentially just get the transcription levels of their genes. And both of these data sets have been increasing at an exponential pace. And I mean that in the technical sense of the word exponential. So on the left is what is the growth of number of protein sequences in RefSeq. And there are very few things in life that can be exponential for 20 plus years. This is one of them, where it is mostly a straight line on a log plot. And on the right-hand side, this is a slightly dated graph, but the numbers roughly hold up, is the amount of expression data and now single cell data is also going up. So this is, in some sense, what has driven the massive interest in using computational methods in biology, right? Because you have all of this data. The paradox is, even with all of this data, it historically had been tricky to make use of it in ways we would want to. So if you look at the kind of data we have, it turns out if it's not often the kind that will help you answer questions that we want to. So for example, if you look at just the genes and proteins, we had a lot of sequencing data come in, because sequencing, anything you can sort of do via sequencing is cheap and you can do at scale. But if you were interested in, say, protein-protein interactions or structure, or for example, KEGG, which is about function and reactions, that you didn't have a ton of. And the other issue was that a lot of this data, and this is true especially for single cell, is you have data from different protocols, different technologies. You're not entirely sure what conditions it was collected in. The gene names might be off. And so a lot of this data is not of the right kind, and it is unlabeled and unstructured. And in this setting is where biological foundation models came in. And this is essentially something that started off with protein language models, but at this point of time has spread everywhere. And the basic idea here is you essentially build upon some of the ideas that have proven so useful in natural language processing, and you train on massive data sets. And the trick is, instead of training it for a specific task, you just try and learn the structure of the data. And you do that by self-supervised training. And in some cases, it is fairly obvious what you should self-supervise on. So for example, if you're working with protein sequences, it is reasonably obvious that you should be using, trying to predict certain amino acids or something. But for example, if you're working with single cell data, it is not at all obvious what you should be supervising on. And in fact, some of the most innovative work that made single cell foundation models possible is thinking hard about what is the right thing to be setting as the loss function. So for example, should you first bin your values and then use the binned values, which will help you deal with batch issues and so on. But if you can do that, you end up learning something about the data. And then you can use that zero shot, which is something that people like to do a lot. Or you can fine tune for downstream tasks, which is how actually most things end up being. Now, in general, and this is partly because in outside of biology, these foundation models have been most powerful for a generative task, whether it is Nano Banana or ChatGPT or something, just creating new content. And that is for sure a valuable application. And I'll show you one application of that. But I would argue that in the long run, it's the representation learning aspect of these foundation models that's going to be more powerful for biology than the generative aspect. And so what kinds of foundation models am I talking about? Basically, at this point of time, there are a bazillion foundation models. Protein language models are the oldest, and they have in general been the most successful, and that's where you see the greatest diversity. DNA language models, RNA language models, and of course, then as you go on, you get to single cell foundation models, you get to pathology foundation models, and now you also are starting to see chromatin accessibility and genome structure foundation models, right? And it seems, if I was to put my skeptical hat on, training a foundation model is a good way of getting publicity and likely a paper in a strong journal, which of course brings in skepticism. Essentially, I don't mean to pick on Anshul at all. I respect him a lot, but his is an opinion that sort of, for example, is reflective in some ways that there can be too much hype in the field. And there is always a steady stream of papers saying that this doesn't work or that doesn't work. And these are all good things because we should be skeptical. So I guess the question is, where does this lie overall? And to this, what I would sort of go back to is a story that came out in the New Yorker about the time when ChatGPT was taking off. And so this was by Ted Chiang, who, if you guys haven't read, is a fantastic writer. And he took, at that point of time, what I would argue is sort of the the more considered but also skeptical view of what ChatGPT was bringing, which was essentially that it is summarizing the web in a way that is interesting. And the idea being that just like JPEGs can essentially take this very high resolution view of the world, figure out just what the human eye can distinguish, and use that to compress it down into a meaningful way. That is what ChatGPT is doing essentially. It is taking this and making it into a sort of a compressed representation of the world. And he came to this from a perspective of essentially saying this is not a big deal. And in some ways, I want to lean into that a bit. So if you think in terms of what this means for biofoundation models, the strong claim for biofoundation models would be that biofoundation models can discover emergent biology, biology that nature never thought of. And this is where often you would go with design, for example, right? But there is a weaker claim, which is the bloody JPEG version, which is that biofoundation models offer you a summary representation of the entire biological knowledge base, or at least some part of the knowledge base. And one argument I want to make in this talk is that as claims go, even this weak claim is a fantastically powerful claim. And if you think about it from that perspective, you can do a lot of things with biological foundation models without necessarily thinking that I need to see an emergent property. They do emerge. I mean, I'll talk about that as well, but there's a lot here. So that sort of motivates the question, which is, how do you discover biology with these foundation models? And so I'm going to tell you about some projects we have done. And I'm going to sort of share with you some strategies that I think are repeatable and that would work across different problem areas in trying to get foundation models and to interrogate them to reveal their biology. So I would sort of pose them as three strategies. The first is identify latent similarities. The second is lean into their representations that might match evolution and nature in ways which we just got lucky for. And the third is to be able to, they help us step back and be able to see the forest for the trees. And so I'm going to go through each of these and give you a few vignettes as we go along. So if I look at the first one, which is identifying latent similarities, the hypothesis here is that many biomolecular properties reside on a continuum rather than as discrete categories. And this is, I guess, one of those things where in biology, when you start by cataloging data and collecting experiments, it's easiest to sort of see the differences and sort of categorize things as, oh, this is this kind of a protein and it's that kind of a protein. But under the hood, it's all biophysics, right? And so more things reside on a continuum than you kind of appreciate from the top. And these models let you get into that. And in particular, about 20% of the human genome belongs to categories of genes, where all the genes in that category have the same broad function, but with different specificities. So this is an analogy I like to give to my fellow computer scientists sometimes, is I like to think of this cellular system as a bunch of dumb motors with a collection of very smart switches. Essentially, the polymerase is a thing that will go do the same thing in every gene. RNA polymerase will do transcription everywhere. It's the transcription factors that decide what gets turned on, right? And so, for example, if you think about kinases, as I'll get to it, phosphorylation is a signal that can be read by a bunch of other entities. but figuring out what gets phosphorylated and where is where you can put in information. So these switches end up being super powerful. And it turns out about 20% of the human genome can be thought of as essentially switches that are of one category that do the same thing, but with different specificities. And so with foundation models, what you can do is you can essentially take these entities and organize them in a low dimensional interpretable manifold. And the hypothesis is that in a low dimensional manifold like this, the continuum will become evident, and you will be able to take advantage of that and be able to make predictions of for specificities that you don't know, and be able to see linkages which were not evident to you before. So this is something we did with our work, Colossus, which is in collaboration with the Scott Soderling lab. And the idea here is we wanted to build a method that can predict kinase specificity across the entire human kinome. And this is obviously an important problem. Kinases are the second most popular drug target. And yet 80% of human kinases have 25 or fewer known substrates. And so we went for a model which basically starts with protein language model representations of kinase sequences and substrate fragments. And it's in this model, we emphasized interpretability. So essentially the idea is we wanted to co-embed the kinase and the substrate in the same space and supervise on them being co-located or in the same point if the kinase substrates phosphorylates the substrate and otherwise no, right? And the work here is a lot, both in terms of sort of making sure your model is robust and something that can generalize, because that is what you're going for here. You have to have a space that generalizes well. That's how you will get the learning and the insights. And so in particular, when we did this, we built a model that can span all kinds of kinases, serine, threonine, as well as tyrosine. But there's been great work in the field already. So Phosphomer ST from the Kannan lab that focuses simply on serine threonine kinases. And they also actually use the protein language model approach. And one thing that we emphasized with our model was, like I said, sort of interpretability and generalizability. And so what we see is that while on overall top level metrics, like the area under curve type metrics, Colossus outperforms Phosphomer a bit, Colossus is substantially more consistent across families. And this matters. And this is something that is an important consideration about model calibration that I think is not given enough importance in the field, where imagine you have two kinases, and for each kinase, given any substrate, you are completely accurate in ranking how much that kinase is likely to phosphorylate that substrate. But it so happens that for kinase 1, your range of value spans from 0.9 to 0.7. For kinase 2, your range of value spans from 0.3 to 0.1. And within 0.9 to 0.7 or 0.3 to 0.1, you are completely accurate in your ranking. But if I gave you a random substrate and I said which one of these is, which of these kinases is going to phosphorylate it, you will incorrectly tell me kinase 1 every single time. And so it's important that your model scores are well calibrated so that if you go to unseen kinases and in vivo systems, you can make confident predictions. And the other part that goes with a method like Colossus is that you have to combine it with the right experiments. So in our case, for example, what we are able to do is say, if this kinase and this potential substrate are in the same place, the kinase would phosphorylate this. What we cannot predict is, will this kinase and this substrate be in the same place? So that is something that Colossus does by pairing it up with an experiment of proximity phosphoproteomics. And so we use that to study sleep. And this was one of the things you learn when you collaborate. I did not know there were genes which basically just make you more likely to sleep or less likely to sleep. That is just a fascinating area. And so on this, we essentially, so SIC3 is the gene. and mutations in it are associated with greater non-REM sleep. And we validated Colossus on our predictions. And the thing that sort of is useful here is that our prediction scores are well calibrated across both in terms of how strongly something gets phosphorylated and whether it gets phosphorylated or not. And then from there, you can build networks of phosphorylation, and you can sort of see, okay, these are the other substrates that are nearby, and those give you candidates to study. But going beyond that, if I go back to the interpretability question, one thing you can immediately do is if you have an interpretable space, if you have a manifold, you can start to lay things out. And you ask, does known biology reappear here? So for example, one thing I'm showing you in these figures is serine threonine versus tyrosine kinases. So if you don't know, what this basically means is there are kinases that typically will phosphorylate a serine or a threonine, and they sort of have interchangeable preferences. And there are other kinases that will phosphorylate tyrosines. And there is a small category of what are known to be dual kinases. And one thing that, for example, turns out if you look at a model like this is that it seems to suggest that, yes, tyrosine kinases are slightly separate, but that the boundary is not quite as stark as you would think. And this is something that we're very interested in, and in just some early experiments for a kinase IRAC4, where our model predicts that this should be a dual kinase, but it has not been reported to be a dual kinase. We are seeing some in vitro evidence that it is indeed actually being able to phosphorylate both tyrosines and serine threonines. It's thought to be a serine threonine kinase only. A second question you can ask is, how functionally accurate is the phylogenetic view of kinases? So kinases are grouped into families, primarily from a phylogenetic perspective. And the understanding is that these families sort of have similar preferences and so on. So you could take your embeddings and you can ask, do these embeddings agree with the phylogeny? And interestingly, for the most part, they do. The place where they disagree are in a couple of families, which are some of the older families where there is known to be some divergence. So for example, here, I'm just sort of zooming into one of these, where say the CAMK and the STE family kind of are mixed along with the TKL family. And this is the kind of thing that you could do simply by just starting with data. That is, yes. And I want to, okay, I had a slide here that got lost. And before I go on to the next thing, I sort of want to make a pitch to trainees in this talk, which is you can do this with a wide variety of gene families. You can do this with ubiquitinases, you can do this with debuquitinases, you can do this with proteases, you can do this with VEGFs, you can do this with histone modifiers, you can do this with GPCRs, you can do this with, there's a few others that I'm forgetting. Oh, for example, chaperones, you can do this with cytochrome 450s. There's a whole bunch of categories where you have proteins that do basically sort of very similar functions, but with different specificities. And you can try to decode that. And in fact, you can do this even beyond at a single protein level. Like I said, we're interested in explaining, but every once in a while, you can go into controlling. And one place where we have been interested in is small molecule modulation of proteins. Can we come up with small molecules that would let you modulate proteins? And obviously, this has drug discovery implications. Here, we approach the problem from essentially a high throughput end. So if you think about protein small molecule binding problem, you can think of the collection of methods as a Pareto frontier, where you are at one axis is mechanism and one axis is speed. So for example, doing a free energy perturbation would be something that is extremely mechanistic, extremely detailed, but extremely slow. What we wanted to do was something that is accurate, but very fast. And the way we went about this is essentially by doing, again, a co-embedding approach, in this case, using protein language model representations of proteins and Smiles representations that converted to Morgan fingerprints, and co-embedding them in the same space. And again, for interpretability and for generalizability, you want to do this carefully. So one thing we did was we actually did this via contrastive learning. So we took examples of decoys, which is small molecules that don't bind, and we required the model to learn a space where the decoys were far away. But what you ultimately end up getting is, again, just a co-embedding space where this works. And what this model is able to do is it's fast and it's accurate. It doesn't tell you where something will bind, but it is actually pretty good at being able to tell you whether or not it will bind. And we did this in a prospective evaluation where we basically took about 19 predictions, sent them out to the lab, and 13 of them came back positive, and I think 6 of them was nanomolar efficacy, and there was one that was not known at all. And which is great, right? Essentially, this is something that you could use at the top of your virtual screening funnel and essentially look through tens of thousands of compounds. But it's not just the speed that helps. It can also give you some insight. So in particular, you can start to ask questions about how are proteins organized in this co-embedding space. And if you start zooming in, right, one thing you see, for example, is a bunch of receptor tyrosine kinases end up in the same place. And so here, for example, what the model is saying is that FLT3 and KIT are very closely situated in the embedding space. And the practical implication of this is that you should expect a fair amount of drug crosstalk between FLT3 and KIT. Like if you make something that is intended for KIT, it might also hit FLT3 and to some extent EGFR, but less extent. And that is again an interpretability thing that you just get out of it. More recently, we have taken this and leaning more into the drug discovery side. We said we want to be able to screen not just 10,000 compounds or 100,000 compounds, but 1 billion compounds. We want to be able to sort of essentially screen these large libraries, which can let you synthesize any molecule on demand from a catalog of like 20, 30 billion listings. And here we ended up doing something that has been again quite successful in the field, which is trying to discretize continuous representation. So in our case, we ended up building a code book of drugs, essentially saying that the entire virtually infinite set of small molecules could be essentially grouped into 32,000 categories. And we did that into a hierarchical tree-shaped grouping, which is 16 times 16 times 16. No, sorry, 32 times 32 times 32. And it turns out that if you put this inductive bias that protein small molecule interaction has to happen through protein code interaction. So essentially you're saying a whole bunch of small molecules interact with the protein through this one mechanism. It actually works quite well. And because it regularizes the problem, it actually ends up performing as well, if not better, than baseline methods. Here's that slide I was thinking of. Yes, so there's a few more things you could work on. You could work on lectins, you could work on transporters, you could work on addition proteins. These are all cases where you can essentially just take some existing data and you can say, can I build a model that predicts specificity across all of them? And would be a solid contribution to the field. Okay, so this is one approach, right, to using biological foundation models to make discovery. A second approach is something where it becomes possible because we got lucky. And we got lucky in the sense that for certain categories of foundation models, specifically protein language models, but to some extent also genomic language models, the way they are trained, the loss functions they are used to train on, seem to mimic, to some extent, evolutionary fitness. So for example, if you think of how a natural language processing model would be trained, it would essentially, in perplexity terms, say that the second sentence, the fish crossing the road, is less likely because the fish does not fit in when I look at the other words on that sentence. It makes more sense to say the chicken crossed the road, right? And an NLP model will think of this purely in terms of statistical frequencies and calling it perplexity, right? One of the really cool things about PLMs is that if you take the same logic and you say, okay, are there sequence substitutions that don't quite work? It turns out that this ends up having actual evolutionary semantics. In particular, mutations that don't quite work turn out to be more pathogenic than otherwise. And this is work that Nadav Brandes has done. It has also something that Brian He did early on. And these are ideas where essentially you're saying the model's training function process itself is somehow so in sync with how evolution learns that I can actually interrogate the model itself to do something. So along these lines, one thing we have been interested in is if we could use PLMs to study dynamics. And this is very attractive because most of us now sort of are seeing that AlphaFold, even as it is an extremely powerful thing, doesn't quite solve the problem because you have to model the flexibility and the dynamics of proteins in isolation, but also in complex. And there's been, and the reason for using PLMs here essentially is the hypothesis that to the extent that dynamics is crucial to a protein's function, evolution should be encoding that in terms of couplings between amino acids. And you should be able to read those off. And the question is, how do you read those off? And so, for example, other work in this area is from, say, Hannah Wayman Steele and Gina Al-Nasser, where they built Dyna. And they essentially sort of used a PLM to try and predict particular kinds of slower range flexibility in a structure. For us, we were interested in seeing if we could predict allostery. And we started with almost a maximalist hypothesis, which is to say that if you look at the attention mechanism in PLMs, the attention mechanism directly should be picking up on allostery. And so the strong version of this hypothesis is that if I have proteins where I know there are two allosteric sites, X2 and X5, I should be able to use that to essentially learn over time with enough allosteric examples which attention heads in a PLM actually are attending to allostery. And we used this. I mean, ultimately, it turns out that this is not sufficient by itself. You want to use the general PLM features too to get more power. But it does give you an insight that, okay, and one thing we found, for example, is that in ESM2, 650 million, the attention heads that sort of score the highest are the attention heads at the later layers, which is kind of nice because it sort of agrees with this intuition that in these PLMs, some of the lower layers are picking up on basic structure while the higher layers are picking up on sort of something more complex. But yeah, and so we built a method allo, which basically just does this. It given a protein sequence, it will try to predict what are the likely allosteric sites. Now, it turns out that it's actually pretty hard to in silico establish allostery because there are very few data sets. So one thing we did was to essentially say, if allostery is, because allostery is functionally important, if I look at cell surface proteins where allostery, where I find, where the residues that I predict to be allosteric should likely be functionally critical. And so if they're functionally critical, then their variant should be pathogenic. So there should be a link between prediction of allostery and pathogenicity on cell surface proteins. And we did that, and it does seem to hold true to a significant degree. So this is something we're still working on, and we are working on improving this method, sort of getting even more confidence in our predictions. But I'll now tell you a different story in terms of how these models and what they learn syncs up with how evolution operates on proteins. So if you think about the sequence function relationship, I'm skipping structure in here, but the idea is ultimately sequence is how all information is passed from organism to organism. So anything about function has to be in the sequence. If you think about the protein sequence function relationship, um, from the functional aspect, the protein is a single functional entity that is doing something. But there's this dichotomy that evolution operates on the sequence. It can make substitution to it, insertions and deletions, and basically it ties out a bunch of different things and ultimately says, oh, this protein does the same thing as that, what I wanted it to do. So this idea that evolution selects for the function but operates on the sequence. One thing that's missing, if you look at how current PLMs behave, is for the most part, if you're given us much of the work of PLMs in over the last 4 or 5 years has focused on using substitutions if you're looking at residue level information, simply because that is much more amenable to mathematical processing than, for example, if you were to argue about, well, how do I handle these two deletions or insertions? Like the way you would typically handle deletions or insertions is to basically compress the entire sequence into a shorter representation. But those are typically useful for discriminative tasks, but not necessarily for generative tasks. So we don't really have a PLM or any other approach easily to sort of think about generative tasks which go across lens while preserving function. And if you could do this, right, there is a whole host of things that you could unlock from a capability perspective. But also, this is from an understanding perspective, super valuable because it sort of lets you understand this question as to what is it that evolution is really trying to protect in a function? What is it that makes it keep a protein like this and basically get rid of irrelevant things or sort of allow for new things? And from a control perspective, if you are able to essentially change the length of a protein, add insertions and deletions without changing other parts of the function, then that gives you enormous capabilities. You can do things like you can make loops shorter or longer. You can make gene therapy more feasible because one of the big bottlenecks in gene therapy is the capacity of the cargo vehicles we use, whether it is AAVs or lentivirals or LNPs. You can make better reporters because they will not be quite as bulky, and all kinds of other things. And we did this with a method we call RECOMB, which lets you essentially generate new proteins given a template sequence. And these new proteins can have uh, a mix of substitutions, insertions, and deletions that you specify. So this is sort of Reagan in play. What I'm showing you here are 3 examples at different points of the length spectrum. So starting from about 300 to 2500. And in each case, we start with the template sequence. And the model does not know anything about what the protein does or how it looks. The model is reasoning entirely just from the data and effectively just evolutionarily. And you tell the model, I want a target length of, say, given CCR1, I want a target length of 464. And I want this level of sequence variability. And the sequence variability can start from 0, where you say, I want very minimal sequence variability from what my template is, to all the way to like whatever you want. But typically at max out around 2 or so. After that, it starts to get a bit all over the place. And here what you see is that it is able to generate sequences, which if you fold them with something like AlphaFold, they end up adopting structures that look remarkably similar to what the original template is. And so a funny story on this preprint came out last year, and it came out about 2 months after the Summer Olympics. And in the Summer Olympics, there was this infamous lady from Australia who was a break dancer, and her stage name was Reagan. And somebody pointed out that our mTOR, actually it was Ann Carpenter, mTOR display looks a lot like somebody breakdancing, and this was completely unintentional, and the name was completely driven by sci-fi and not by the Olympics. But yeah, it's good, fun. So what's happening under the hood is, so I'll just, under the hood, what it is doing, so this is an example with hemoglobin, is it's just sort of reasoning about where it can make insertions and substitutions and changes and deletions. So essentially, when you specify that I want to make it to be this long versus that long, it doesn't sort of randomly go and add things to the ends. It will figure out what are the places where there is give. And under the hood, this is implemented through an autoencoder architecture. So the key insight is that we created a shape for the latent space, which we thought has enough capacity to capture the overall fold structure and the function of the protein, and yet be essentially something that you could encode to and decode from. And so we use the ESM2 based model, and we sort of simplify the problem by saying my goal is actually to go from ESM representations to ESM representations, because it turns out decoding ESM representations to sequences can be done with high confidence. So you start from ESM, you go to ESM, you learn this encoder, which is a collection of transformer and neural net and CNN blocks. And then you essentially, the latent space is, in the current implementation of RECOMB, 50 contiguous blocks. And effectively, sequences sort of get locked down into these 50. And when you want to expand, you essentially just sort of pull out from there as to whatever length you want. And the whole idea is you do this in a self-supervised way. So essentially, self-supervision is again coming back here. And the other thing that happens is, as you compress, because you are averaging at a point, you can lean into the central limit theorem and say, when I take averages like this, I can also get a standard deviation estimate, and my average will actually be likely to behave like a Gaussian. And that makes for a very natural sampling approach. So unlike sort of diffusion, where you essentially are following a trajectory of noise from here to there, in RECOMB, the idea is that you map to a point in the latent space, and you actually get not just the point, but a Gaussian distribution around the point. And you can just sample from that Gaussian distribution, and you literally specify essentially the multiplier on the sigma. If you specify a 0, you literally get the same point back. If you specify a higher multiplier, then you essentially get further away from the thing, and you just decode it to what length you want. And of course, there's a whole bunch of other detail that goes along with this in terms of how you should filter it and so on and so forth. And all of that is in the preprint. But, oh, actually, I did went through this slide. Yeah, so this is the one I was talking about, where essentially you don't have to use diffusion, but you can go through. And you can then start to use this and say, how well can you miniaturize or magnify sequences? And what we find, and this is essentially a property of nature, right? Like this is something saying, if I take a natural protein, what are the odds that it has some give? And what we find is that up to 5 to 10%, you can essentially, for most proteins, you can either remove 5 to 10% of amino acids or add 5 to 10% of amino acids and still maintain relatively good PLDDT. And also, in general, maintain high alignment, structural alignment with the original structure. You can, of course, ask RECOMB to do much larger deletions. And the interesting question then becomes is that what kind of deletions does it choose to make? And I'll tell you a story about that like in a little bit. But just to show an example of what you can do with this. So one direction that we have gotten very interested in are biosensors. From a control and again discovery perspective, biosensors are fundamental to a lot of biology, but also diagnostics. And I think we haven't scratched the surface of what AI can make possible with biosensing in terms of all the new kinds of sensing that we can do. And so we actually posed for ourselves like a simpler problem, which is saying, can I simply miniaturize some of these fluorescent proteins? And these fluorescent proteins, they are small, but there are many human proteins that are smaller. So it would be good to have small fluorescent proteins. And so here we essentially took green fluorescent protein and red fluorescent protein, and we built variants of this. So the one on the far right, get what is the pointer, is essentially the first fluorescent protein that is sub 200 and cofactor free. And this is one of those things where you have to take structured prediction with a little bit of um, with a bit of a grain of salt, because some things can look like they don't fold well, but if the language model likelihood is good, they often end up expressing in the cell. And this is something that Adaptive Bio found in their contests when they were trying to do a contest of EGFR binder design. In the first round, they found that things didn't express quite as well. In the second round, they added in a PLM-based PLL filtering, and that seemed to help with that. So we designed these, we synthesized these, we tested them in vivo, and they work. I mean, 5 out of the 8 of those work. Sorry. And this is sort of the other side of the coin in terms of the sequence function landscape, which is they work, but they're not quite as bright. And this is an open problem, which is figuring out how do you get higher levels and ideally maybe even supernatural levels of function. That is something that evolution might not have encoded. And those are really interesting representation learning problems to figure out. How do you run these representations where you can get the kind of function that you want? And I told you a story about looking at what Reagan can do if you make it shrink things by a crazy amount. So we tried this with biotin ligase. Biotin ligase is essentially attach biotin to proteins near them, and which you can then pull down. So it is a good way of doing proximity assays. And there are two bread and butter reagents in this. One is Turbo ID, which is about 300 odd long. And there is another called Ultra ID, which is about 160 or 170 long. And it was made by trial and error from Turbo ID by people just sort of fiddling with it and saying, okay, what domains can I take out? And we asked Reagan to essentially shrink it to roughly the same length as Ultra ID, 165. And interestingly, it automatically, without knowing what Turbo ID is or anything about its structure, it chose to remove that domain which human experts had removed on their own after a fair bit of tinkering. So that's a good part. And when you go and express these, a fair number of these express. But again, their function is weaker than we would want it to be. So that's the thing we're working on. You can also use this to expand things. So we actually participated, Youngsu, who's in the talk here, built an EGFR binder based on EGF. And I think in the second round of the adaptive biocomputation, it was probably one of the better EGF-derived binders. So that was cool. Okay, I'm running out of time, so I'm going to go to the third strategy, which is foundation models let you see the forest for the trees. And the idea here is sometimes patterns are not visible to you when you look at it one data set at a time. You have to be able to do something where you put all the data sets together, and only then the patterns are visible to you. So in particular, this is where I now switch to talking about single cell foundation models. So the question that we're interested in here, and it is a fascinating question if you start thinking about it even for a little bit, is how do we have the same fixed genomic ordering that is able to encode these very diverse cell types, which means very different gene combinations of expression? And the natural answer is that this is done by using the 3D genome. There are parts of the genome that fold away in certain cell types, so you don't get access to them. There are other parts that are accessible. And the story goes here that there are regions like topologically associating domains where you can essentially sort of group genes. Good, but this doesn't fully explain things, Because one thing that turns out is these boundaries are more stable than you would have thought. And in fact, not just across cell types, but to some extent across species as well. So then you go back to the same question as to how is it that they help? And if you look in nature, bacteria have a much simpler solution to this problem. They lean into the biophysics. They basically essentially have operons where if you have genes that are doing the same thing, they end up being on the same plants, right? But bacteria have it easy. They have to do just one thing. I mean, we have to do so many things. I'm sorry, just making a joke. So the thing is, okay, this 3D structure exists, and the claim is that this 3D structure is important in controlling genes. But can we show that? Thank you. Sorry about that. So essentially, the state in the field is there's a debate about how much stats really matter. And so if you go and look at, for example, expression of genes that are sort of in the same TAD, you actually do find that they are co-expressed. It's not very significant, but genes that are in the same TAD are more co-expressed than genes that are not in the same TAD. But this is confounded by the fact that just along the genome, genomic proximity corresponds to sort of some co-expression. So it's not clear if the TAD organization is actually making a difference. So the first thing we did, and I'll skip through that part, which is essentially we said, let's actually collect TAD data from a bunch of different cell types, and let's compute a consensus TAD structure. This is through dynamic programming, all of that, ignore it. But at the end of it, what you have is for the human species or for a mouse species, you have a delineation of the human genome across. This is where TADs are typically formed. And now this is where the forest and the trees analogy comes in. One way of computing expression similarity between genes is to directly compute correlation. So you have a collection of cells on which you have genes. So this is, for example, any single cell data matrix, and you compute correlation. A second approach is to essentially first take that data matrix or millions of such data matrices, plug them through a self-supervised learner, and then look at the gene embeddings you have and look at similarity between those gene embeddings. You would think these should be picking up the same information, right? Turns out they don't. So this is what I'm showing you here. And this is the trees versus the forest figure. So the x-axis is co-expression when you compute it pairs of genes at a time on single cell data sets. And this is across a lot of single cell data. So it's not like the data is the issue here. And the y-axis is essentially you train a model first, then you look at the gene embeddings, and you get the correlation between the gene embeddings. And if you look at it, what you will notice is that they actually kind of agree. I mean, there is a XY line you can sort of imagine through this, but there is quite a bit of disagreement. And in particular, there are many, many gene pairs where the co-expression level is practically zero. And the contextual similarity can take on wide ranges of values. And part of this is that single cell data sets are noisy. And even if you have millions of data sets on any two single cell transcriptomes, you only have some overlap. So you'll have muted levels of transcription. But there's also something deeper that happens. So these are two particular examples where for two gene families, what you notice is that if you were to compute expression in sort of the trees way by just looking at pairs of genes, you almost always get a zero. While if you look at, for example, the expression of what the contextual similarity or the foundation model similarity, you get substantially higher values. And the interesting thing about these two genes is that they are essentially families of genes that again play the same role, but are typically mutually exclusive. For example, an olfactory neuron is unlikely to express all olfactory receptors at the same time. Typically, it'll express one. So there is a thing where if you look at two olfactory receptors, they don't play they don't show up at the same time. But to the rest of the cell, those two entities are playing the same role. So their foundation model embeddings end up in the same place, and you end up seeing a higher correlation. And what this buys you now is two things. One, it's giving you higher data quality, so your analysis can be better powered. And two, it is actually revealing something that you might not have appreciated, which is that there are these categories of genes. And so, for example, you see this for some histones as well and keratins, where certain categories of genes are essentially just, they have higher contextual similarity than you would have expression similarity. And if you go and apply this, you start to see that, yes, TADs indeed have a higher expression level. And in fact, they have a higher expression level than what you would expect by just genomic similarity. And in the preprint, we actually went further out and we sort of tried to speculate why this might be happening. And we think this has to do with transcriptional condensates, where we think, and this is a hypothesis, there is a synergy between TAD formation and condensate formation. So it's not simply that TADs bring genes together, because if that was the only reason, you would see some patterns of distance-dependent decay that we do not. Instead, what we see is that in TADs, even if genes are far away, you get a bump in the level of similarity. And that makes more sense if what TADs do is essentially make it easier for condensates to form, so that any pairs of genes you can see more. And we, so the we as in my student Juan did this really cool analysis where he essentially took perturb-seq data and asked a very different question from it than people typically do. So people typically, they look at perturb-seq data and say, what is the impact of this transcription factor? Instead, he said, let me take this transcription factor and look at all the all the changes that happen, and essentially compute the changes that happen on genes that live inside TADs and genes that live on their own outside TADs, and compute essentially the impact in terms of the fold change in expression computed by DE-seq. And can I group transcription factors by ones which have more impact on genes inside TADs and other or chromatin remodelers that have impact on genes outside? And we find that there is a pretty interesting delineation between these two categories. And some of the condensate relevant genes show up in this red bucket, which are the genes that seem to have more impact on TADs. And we also went and looked at some cancer data, and we found that this also has an impact on cancerous versus normal cells, and also when you actually try and treat the cancer. I know I'm past the time, so I'm going to stop here. I want to give a shout out to people in the lab. All of this work was led by trainees in the lab from Kapil Dev Kota, Huan Liang, Mert Erden, and others. And we're recruiting, so please reach out. Want to thank our funders, and happy to take questions.
Awesome. Thanks so much. So I gave everybody who stuck around permissions to unmute if they want to ask a question. I will read off one question that came through the chat, and then I'll give some time for people to unmute if they have some additional questions. So Calvin in the chat wrote, in addition to using raygun to modify sequence length, would it be straightforward to use the same overall approach to modify other properties such as thermostability?
It's a really good question, and we are very interested in doing that. So we see it as a general generation platform on which you can apply other styles. And so, yes, please reach out to us, and we're very interested to talk more.
Awesome. Another question that came through from Gabriel says, do you speculate that synthetic TAD creation may be a possible alternative to CRISPR-A?
Oh, that's just a creative idea. Yeah, I mean, yes, that sounds at the moment, I can't say why this should not be possible, but at the same time, obviously, there's a whole bunch of things you have to think through in terms of getting the right CTCFs. If you wanted to go that way, one thing I would suggest is maybe think about combining some of the genomic language models with work that has, for example, people like Katie Pollard and others have done in predicting genome structure from sequence, and see if your genome changes lead to that TAD formation, and then go for it.
Awesome. Another question that came through from Firkin asks, do you always use the 650 million model for ESM2 embeddings? What determines your choice in your ESM2 size?
This is a great question. So Colossus, we actually ended up using the 15 billion model. In general, we have found the ESM2 650 million model to work quite well. That said, PLM scaling is a problem we are quite interested in. And for example, one of the things that's holding back the field is that we can't reliably scale up these things. And even as people are doing substantial progress, make substantial progress in, for example, new kinds of foundation models, multimodal foundation models, 100 billion parameter models, discrete diffusion models. The lift in terms of function or prediction hasn't been so high that the field has moved on. And I think it's important to think about how we can do that. That's definitely a question to ponder.
Awesome. Another question that came through from Land says, for the comparison with the FOMER model, why
Was
Colossal
former,
S
yeah. No, it's
performs
okay.
better than any specific kinase family? Both are based on the ESM language model, so what makes the difference between the two?
Yes, that's a good question. And I think that sort of is what I was trying to get at when I was saying think about latent spaces is uh, we went for an approach where essentially the architecture makes the space very heavily regularized. It puts fairly large constraints on what the co-embedding space would be. And that gives you some power in sort of being able to say, I will have better performance or more stable performance. And that's my point about that whole strategy. If you're going the latent space, you have to think carefully about how you create that latent space such that you can use it for interpretability, because that is the most powerful thing out of it.
Awesome. And then one follow-up question that I had from someone on our team today that I don't think you covered, maybe I missed it, but speaking on behalf of interpretability, what do you think about some of these? I think most of the field has moved away from things like explainable AI, but people have started exploring with things like sparse autoencoders or things like that. Do you have any thoughts about that for interpretability? I guess that would be more post hoc or other things like that that you find interesting.
I think those directions are all worth exploring. SAEs are great in the sense that what they show is these models have concepts in them that can be mapped to biology. Uh, but that, how do you make use of it as opposed to just sort of being able to say, yes, this exists, is unclear, because SAEs impose a pretty heavy ask on the user in terms of being able to annotate things and saying this is what it is and so on. I think that's one of the missing pieces here. But they're great. I think with our approach to this has been historically sort of problem oriented, where we say we want to get the interpretability for the problem specific thing as opposed to a more generic version of interpretability. But yeah, I think it's all of the above.
Awesome.
Like do
And then one more question that just came through, also from Firkin, was, do you think data augmentation is a feasible idea to break out of the inverse scaling trend in the protein language model scene?
I Yes, but you have to be careful. So if you think about what makes PLM specially useful is us being able to sort of like us benefiting from evolutionary rules that mimic how we train things. If your synthetic data does not adhere to that, then it's not clear to me what you're learning. So for example, an analogy would be on the single cell side. Um, there isn't necessarily a particular rule that these single cell foundation models are learning, right? And you could think of, okay, their synthetic data might be a great thing to do as well, but also there you're getting a lot more data. So synthetic is great, but if your distributional assumptions are not lining up, then you might be signing up for something that you might not anticipate.
Awesome. Well, I want to thank everybody who attended, especially those who stuck it out through the end to hear all the Q&A. Thank you so much again, Rohit, for giving us a talk today, and we will see everyone in the community at our next event. Thank you, guys.
Thank you so much. Bye bye.